My name is Michael Choo. I am the co-founder and CEO of Choo Therapeutics. And on the call with me is Dr. Mary Garretson, our Chief Scientific Officer. Quickly about us, Choo Therapeutics is a preclinical stage biotech company at the forefront of developing peptide drug conjugates for kidney and urinary tract diseases. Our technology, UroPep, is a novel, non-invasive, selective drug delivery system that targets the urothelium. It was developed at Weill Cornell. It's a platform technology that would reduce repeated surgeries and replace surgical installations of drug therapies. Our lead candidate has shown significant reductions in tumor growth and survival benefits in urothelial carcinoma. We have a strong connection to Cornell. My co-founder, our chairman and co-inventor, Dr. Benjamin Choi, on the top left hand, is a clinical assistant professor in urology at Weill Cornell. As also Dr. Vanessa Bollé, Dr. Benedict Law are professors in the radiology department at Weill Cornell. And Dr. Scher is a professor of urology at Weill Cornell. So despite the fact that the kidneys are one of the most vascular organs in the body, they function as a filtering organ to eliminate body fluid waste, thus blocking essential elements such as protein, blood cells, and antibodies from reaching the urinary system. Therefore, targeted therapeutic drug delivery to the urinary system has been unattainable via systemic circulation. Due to these physiologic barriers, doctors have to use catheters into the bladder or surgical instruments to deliver therapies directly into the bladder. For instance, upper tract urothelial carcinoma of the kidney lining and the ureter has a high unmet need, as the current standard of care is surgical in nature, with 95% of all patients undergoing a surgical intervention, 80% having kidney removal in order to treat upper tract disease. Our solution is a novel peptide drug conjugate platform to systemically deliver drug therapies to the urothelium without off-target distribution. Our solution would be the first and only non-invasive systemic administration of drug therapies for these type of diseases. The rapid renal excretion would deliver our payload to the urothelium with limited systemic toxicity. Our technology is based on a proprietary peptide drug conjugate system with UroPep, our technology, as a small negatively charged bioinert peptide that is stable in plasma and urine. It takes advantage of the well-known rapid physiological clearance of peptides by the kidney, and we formulated and designed UroPep to carry the payload to the urinary system. Our lead molecule is a UroPep chemotherapy conjugate consisting of DM1 as a payload, which is the most successful chemotherapy agent attached to antibody drug conjugates, Kadcyla, which is the first antibody drug conjugate that was developed for cancer, for solid tumors. Our animal studies have shown positive results for both biodistribution, survival benefit, and off-target toxicity. Our technology is for a proprietary peptide system that is delivered exclusively through the kidneys. And our pipeline is to address first upper tract urothelial carcinoma, second bladder cancer, and third interstitial cystitis. But our platform is broad enough to attach different types of warheads, including antibiotics, steroids, immunosuppressants, and other drug payloads. Our lead asset, Choo-1001, has gone through some preclinical studies for our first indication, which is upper tract urothelial carcinoma. And our development plan today is we have 1 patent issued and 9 pending. We have built some orthotopic animal models for urothelial carcinoma and renal cell carcinoma, including some non-GLP talks. We've completed some mechanism of action studies and are in the midst of developing our pipeline. And we're here today because we're trying to come up with, we're launching our fundraising efforts for our seed round of financing, which will get us through the IND enabling studies to get our lead asset into the clinic. To date, we have raised about $1.5 million in angel funding, and we have been extremely capital efficient in terms of getting our mechanism of action studies, our preclinical studies for our lead candidate. And the next milestones is completing some PK studies, some non-GLP tox studies for our IND enabling studies, which will get us to our pre-IND meeting, and then to our IND within 14 months of inception. So I guess a lot of information to share with the group in 8 minutes, but let us open the floor for Q&A to the broader group. And please feel free to join us during the networking session from 1:30 to 3 so that we could go through our data in more in depth. So I give the floor to you, Jamie, to.
Thank you, Michael. I'll read questions that come in the Q&A, and if anyone wanted to come to the stage, we'll try and manage that. And just a heads up, Michael and Mary will be at table 7 on the second virtual floor if you want to meet them during the network portion. So Jessica Fletchner asks, have you considered or had an interact meeting with FDA for partial de-risking of the IND enabling path? FDA interact meeting.
We have not scheduled our pre-IND meeting. We have some development milestones and studies. Mary, do you want to talk a little bit about what we anticipate speaking to them in our pre-IND meeting?
Yes, we've done some preliminary tox studies, but they've been in mice. And basically what we want to do is to get sign off from the FDA with regards to our IND plans to use rat and dog as our tox models, and to complete the verification and validation of our PK methodology, as well as scale up of our compound.
As Mary mentioned, we have conducted some non-GLP tox studies and dose range finding studies in normal mice. And we are developing the protocols for rats and dogs.
Great, thank you. If anyone else have questions for the Choo Therapeutics team, feel free to enter them in Q&A or chat or raise your hand.
So we do have quite a bit of data, both from within Cornell and after the launch of Choo Therapeutics. We would love to share with you a little bit about the mechanism of action, the preliminary survival benefit studies, the tox studies, and the platform in terms of mechanism of action. So please join us at table 7 during the networking session on floor 2. We would be happy to share with you some of our results in more detail.
Great. And one last question had come through before we transition to our next team. Has any scale-up work been initiated with a CDMO for GMP production?
We don't have work that has been initiated. We have several quotes in place for scale up as well as a proposed route of synthesis.
Great. Thank you. Well, thank you, Michael and Mary, and hopefully the participants can join you virtually in the networking session. We'll ask you to stop sharing. We'll get ready for our next speaker.
Thank you.